Hello students. In the series of lecture we were discussing about the cytoskeletal system of cell. And in this lecture we will be discussing about the difference in microtubules, microfilaments and intermediate filaments. These are the main three components of the cytoskeleton as we have already studied in the previous lectures. First, just a brief recap of what we have discussed so far in our previous lectures. So what are the components of the cytoskeleton? We studied they are the microtubules, the microfilaments and the intermediate filaments. The microtubules are found in the interior of the cell where they maintain the cell shape by resisting compressive forces. The microfilaments thicken the cortex around the inner edge of the cell and like rubber bands they resist tension, whereas the microfilaments are found throughout the cell and the hold organelles in place. The units we studied in the previous lectures are different. That means that the basic structures are different and they are made up of different proteins. The microtubules we saw are made up of alpha and beta tubulin proteins, and the microfilaments are made up of the G-actin protein. The monomers of the G-actin polymerize into the microfilaments of F-actin, whereas the intermediate filaments are made up of a variety of proteins. There are about 70 different genes coding for various intermediate filament proteins. However, different kinds of intermediate filaments share basic characteristics, and in general, they are all polymers that measure between 9 to 11 nanometer in diameter when they are fully assembled. So let us begin with the differences between the three components of cytoskeleton. First of all, let us see the structural differences between the three components. The microtubules, they are the largest elements of the cytoskeleton, and a microtubule is a hollow cylinder of 13 protofilaments. They are stiff, hollow, inextensible tubes. Whereas the microfilaments, they are the smallest elements of the cytoskeleton. They are flexible, inextensible and helical filaments. Coming to intermediate filaments, these are intermediate between the two, that is between the microtubule and the microfilaments, and they are tough, flexible and extensible. Coming to the dimensions, if we see the microtubules, they have 25 nanometer outer diameter. The microfilaments have 8 nanometer diameter and intermediate filaments have 10 to 12 nanometer diameter. Further, coming to their distribution, the microtubules and the microfilaments both are present in all eukaryotic cells. Intermediate filaments are present on all the animals. Coming to their subcellular distribution, microtubules and microfilaments, they are both present in the cytoplasm, whereas the intermediate filaments are present in the cytoplasm as well as the nucleus. Now let us see what are the subunits incorporated into the polymers. We have GTP alpha beta tubulin heterodimer in microtubules, and we have ATP actin in microfilaments, and we have more than 70 different proteins in intermediate filaments. That is, the alpha beta tubulin binds GTP molecule, the actin binds the ATP molecules, whereas the intermediate filaments do not bind any nucleotides. Now coming to the preferential sites of the incorporation, that is, we will see what are the preferential sites of incorporation of these subunits. The subunits in the microtubules are added or incorporated at the plus end. The subunits in the microfilaments are also added at the plus end, the barbed end, and in the intermediate filaments, the subunits are incorporated internally. Now, let us see the enzymatic activity of these proteins. The microtubules have GTPase activity while the microfilaments have ATPase activity, and the intermediate filaments do not show any enzymatic activity. Moreover, these filaments have polarity with plus and minus ends. Both microtubule and microfilaments have polarity, whereas the intermediate filaments do not have polarity. The important function of cytoskeleton proteins is the part that they play in the motor activity. So, what are the motor proteins? Both microtubules and microfilaments act as a drag for motor proteins. For microtubules, we have kinesins and dyneins. For microfilaments, we have myosin motor proteins, whereas intermediate filaments have no motor proteins. Now let us see the different major proteins associated with the cytoskeleton proteins. There are MAPs proteins, that is the microtubular associated proteins for microtubules. The MAPs act as a stabilizing and bundling proteins. There are actin binding proteins for microfilaments, and we have pleckins for the intermediate filaments. Now, the important difference is in their functions. Microtubules support the intracellular transport and cell organization. The microfilaments take part in the motility and contractility, like muscle contraction through actin myosin, whereas the intermediate filaments are for structural support. So, the cytoskeleton of a cell is made up of microtubules, actin filaments, and intermediate filaments. These structures give the cell its shape and help organize the cell's part. In addition, they provide a basis for the movement and cell division. With this, we come to the end of the cytoskeleton components. And these were the major differences between the microtubule, microfilament, and the intermediate filaments. I hope the lecture was clear to you. In the next class, we will start with another chapter. Thank you.